Good morning, everyone. I hope that everyone is doing well today. Welcome to the March 2021 Impact webinar. My name is Rebecca Bailey, and I will be your moderator today. We do have one housekeeping note. Our speaker today will be taking questions at the end of the webinar. You can ask questions at any time during the webinar by entering the question into the appropriate pane on your GoToWebinar side panel. And again, we'll be addressing all of those questions at the end of the webinar. Today, our speaker is Dr. Amber Smith. Dr. Smith is an assistant professor in the Department of Pediatrics and the Institute for the Study of Host Pathogen Systems at the University of Tennessee Health Sciences Center and Le Bonheur Children's Hospital. She received a PhD in mathematics at the University of Utah and completed postdoctoral training in biophysics and theoretical biology at the Los Alamos National Laboratory and in infectious diseases at St. Jude Children's Research Hospital. Dr. Smith's laboratory integrates computation and experiments to study the pathogenesis of respiratory infections and co-infections. So today, I'd like to welcome our speaker, Dr. Amber Smith. Welcome, Amber.
Thank you so much, and thank you for that introduction. So as Rebecca mentioned, I'm going to be talking today about some of our work in pairing mechanistic mathematical models together with some experimental work to show different things about the progression of the influenza infection and what it actually means for pathogenicity and how that then can relate to looking at efficacy of antivirals. So I'll get my acknowledgements out of the way in the beginning. Most of the work that I'm going to talk about today was done within my lab with my really talented lab manager, Amanda Smith, and an undergraduate student who did a lot of the modeling, Maggie Myers. And then we did this also in collaboration with St. Jude Children's Hospital, and of course we're funded by the NIH and some institutional funds. So when we think about virus infections and why we might want to use computational techniques, including mathematical models, is we're really trying to understand the dynamics of the virus infection, not only to develop effective therapies, but really understand and be able to forecast and predict different aspects of the influenza infection, or just of actually any virus, of any viral infection. Mathematical models have been employed across a wide range of viruses with quite a bit of success in teaching us about how these viruses replicate and grow within a host and transmit to other hosts as well. And so we really try to ask questions such as, how does the virus spread and cause disease? And then how can we perturb those with different types of therapeutics? And then one of my favorite questions is understanding how the processes are altered by different viruses or bacteria. So that's kind of thinking about that infections are sometimes single infections, but a lot of times there are multiple pathogens present and they might be interacting. But for the purposes of today, I'm going to be focusing mostly just on the influenza infection. So I put two representative figures of two different virus infections. On the right-hand side, you'll see HIV as a patient goes through the acute phase and then sort of settles out into the chronic phase, and it's over a much longer time scale compared to influenza, which lasts about 0 to 10 days, maybe at the most, and the viral loads go up really fast, and then it actually clears pretty fast comparatively. And this is true for most viruses. So here's just a few other examples and what dynamic profiles of these virus infections actually look like. And they can really be categorized into about 3, although you'll see 4 on there because there's a chronic infection and then there's the acute infection that actually will clear. So some of these are more of a monophasic decay where they just shoot up really fast and then clear really fast. Some have 2 different phases of decay, and even up to 3 is different aspects of the immune response are kicking in and trying to clear the pathogen. So for influenza in particular, if you're not familiar with influenza, we actually haven't seen much of it this year, thank goodness. But it's an RNA virus that infects the respiratory tract. It infects the upper and lower respiratory tract. A lot of influenza infections are not that severe, where they are limited to infecting different parts of your nasopharynx, and then they never make it to the lung. But once they do make it to the lung, these can be really severe infections. So on a normal year, we have maybe 10 million plus infections and 20,000 plus deaths each year in the US. And that's considered a pretty mild flu year. You can see here on some recent figures from the CDC that about anywhere from 5 to 10% of hospitalizations are actually from influenza or influenza-like illnesses. Now you can see down here in the little red triangles, that's this past season. And that's primarily because we've all been at home. Our kids haven't been in school. We haven't really been traveling as much, and we're wearing masks and social distancing, and all of that has contributed to this unusually low influenza season. But it really does show the power of wearing a mask, washing your hands, and not having as much contact with everybody. And where that has really translated into something that's pretty robust is on the right-hand side. You can see the influenza-associated pediatric deaths. So comparatively to COVID, influenza infects and affects children much more. And so we're a little bit lucky in the sense that COVID has not hit our children as much, but influenza predominantly does. And so these are just pediatric deaths, approximately 150 to 200 a year. And so far this season, we've only recorded 1 single death, which is quite remarkable. So given all the bad things that have happened because of COVID, I guess this is a little bit of a silver lining. And as I mentioned earlier with the secondary bacterial infections, they in past pandemics have actually been the cause of most of the influenza-associated mortality. So in different pandemics, this is anywhere from about 40 to all the way up in the 1918 pandemic to 95% of the mortality. And that's why I really like that question, is because it just increases the complexity and causes so much more morbidity to the population. And of course, everybody knows that there is a vaccine available, although we have to keep changing it because the virus mutates so quickly. And antiviral therapy is still really problematic. We have 2 decent drugs on the market, but neither one of them do a really great job. And I'll talk a little bit about their window of efficacy in just a minute. So here are 2 viral load profiles. This is both in humans, and these are upper respiratory tract. So of course, it's very hard to sample the lower respiratory tract and understand what's going there, but we can sample the upper respiratory tract and look at viral loads. These are 2 different influenza viruses. They're labeled with their surface protein, so hemagglutinin and neuraminidase are the H's and the N's here. So we have an H1N1 and an H3N2, and you can see how their viral loads differ just a little bit, and all the individual lines are different patients. So you can also see how people vary in their disease course and potentially in their severity. So if we think about their symptoms are a little bit hard to gauge. And with humans, there's a whole range of symptoms that are anything from a fever to cough to aches, body aches and chills. So they try to quantify that by using a symptom score, which is a little bit of a tricky thing because people probably gauge that a little bit different. But on average, this is what some of the symptom scores looked like for different viruses. So here's that H1N1, an H3N2, and an H2N2, then influenza B. And all of these are a little bit different, maybe have different time scales, a little bit different dynamics. But one thing's for sure is that they don't exactly look like the viral loads. And one would expect that symptoms might track that. And that's true to an extent, and that's what I'm going to talk a little bit about today. So if we look at this in another light, you can also see that if you look just at, say, sneezing, that will happen much earlier in the infection compared to a cough when it gets into the lower respiratory tract, and yet that might persist for significantly longer. And longer after you have already cleared the infection, you can still have symptoms. And that's really come to light with COVID as well, where people are still experiencing symptoms long after they have had the infection. And this is just a CT image from the 2009 pandemic. What you can see here, these lighter areas are where the infection was or currently is. And you can see that it's on both sides of the lung and this huge area of the lung that has been impacted. So we have a little bit of the benefit of being able to study this much better in animal models, and in particularly in mice. And there's a lot of similarities between mice and humans, including the time scale of infection, what the viral loads look like, the disease course, and the efficacy of antivirals when we put them into these animals. So these are data from my own lab, where we did an H1N1 infection in BALB/c mice. Our viral loads take that biphasic decay where they really flatten out in the middle part of the infection and then clear very rapidly in the last few days of the infection. So for mice have a different symptom profile. They, of course, don't really cough or sneeze, so we can't measure that in that way, but weight loss is a really good indicator of disease severity. And it's what's used across pretty much every study to monitor how severe a disease is going to be. And similar to the humans, you can notice that this weight loss looks almost nothing like these viral loads. But somehow they are obviously related because the virus is causing the disease that is causing the animals to lose weight. And that's kind of what I'm going to talk you through today and take you through this journey of how we can get from viral loads all the way over to disease severity. And the short story is there's connections to what the dynamics of the infected cells look like, different immune responses. We're going to focus on the CD8 T cell response, how those things interact to cause the actual lung injury, which is distinct from inflammation, although they're often used interchangeably. But we'll see today that the dynamics are completely independent, and we can also get from these infected cells to inflammation, and then from both of these things down to disease severity. Okay, so the way that I like to also approach these problems is we really want to have mechanistic models that we have a lot of faith in, that we've potentially validated within the lab so that we know and can be confident of our predictive abilities with looking at some of these things and how the virus grows and is cleared by different immune responses. So the way I usually start a study is developing a model, just establishing feedbacks, testing different hypotheses, or generating new hypotheses that are necessary to fit a piece of data. We usually calibrate our models as long as we have sufficient data to do that, really investigate and estimate different parameters and how those parameters might be related to one another. We do some model analysis, looking at the sensitivity so that we can get an even better understanding of how our model behaves and make some predictions from that. We might be able to look at where the dynamics might bifurcate and cause some heterogeneity. So we perform little in silico experiments to try to understand the system and make predictions. And those predictions can be anything from the regulatory mechanism that is driving the whole system to looking at what happens under a perturbation, such as an antiviral. And then we take these back into the lab, try to design an experiment that can get at whether or not our model is predicting it correctly, identify any inaccuracies or accuracies, and then refine the model and sort of do this again. So if we rewind time just a little bit, some of the first influenza models were developed back in 2006 by this really seminal paper by Alan Perelson's group. And in that paper, they used human data. So this is upper respiratory tract viral loads. They were volunteers that had been infected with influenza. And this is the data that I showed earlier as well. So we have this exponential rise and decay of the virus here. It peaks maybe about 2 to 4 days, and then it clears within about 7. They developed this model, which really came from a modification out of the HIV literature. In this model, we have target cells, which are denoted by T. Those are your, in this case, your epithelial cells. They become infected at this rate beta V. They first undergo an eclipse phase, which is the time that it takes a virus to replicate and begin producing virions. And so once that happens, these I2 cells can produce virus. That virus can then be cleared, or the infected cells can be cleared either by host responses or succumbing to having been infected. And so this black line over here is the fit of this model. On this short time scale, we assume that the target cells, the epithelial cells, are not regenerating on the same time scale. And so we don't have a replenishment. So once they are all infected and are down here near 0, the virus then starts to decline because, of course, there's no new cells to come in and infect. And that's a little bit of an approximation. It's not to say that every cell in the lung gets infected or every cell in the nasopharynx gets infected, but it's a good approximation to use. So some of my early work took this model and actually applied it to animal data. So we did it in mice. The fit looks very similar, and that's because mice are a really good model for human influenza. We came up with a few different rates, but they were very minor comparatively. So some of the other work that we did, and this is really important when thinking about antivirals, is we did a little bit of mathematics to look for approximate solutions so that we could actually solve the system and try to understand which parameters and which processes were acting on which timescales. And so one of the things that we found is this initial virus growth involves all the different processes that I just mentioned that are in this model. You can approximate it with an exponential, but the exponential rate is a whole bunch of parameter combinations. And that actually makes a lot of sense. This is when the virus is really replicating and growing very quickly. And that exponential growth happens for about 2 days. And this is exactly why antivirals that really target some of those virus replication processes will not be as efficacious after this time. So we really have that short 1 to 2 days, especially for influenza, to administer antivirals and have a robust response from that. So after this is about the time when the immune response is starting to kick in. The virus is slowing as it gets to the peak. This was a truly nonlinear phase, so a lot of things are happening at once, and it's at about 12 hours before the peak. And then after the peak, the entire viral decay was really dominated by infected cell clearance. So having your immune system responding and clearing out those infected cells really drove that down. So if we think then about what that means and how those parameters influence these viral load curves, we can understand now how we can model this biphasic decline. And so here are just 2 different data sets that also show that multiple viruses can sometimes have these biphasic viral clearance phases, and that our standard viral kinetic model that we have loved for a very long time could not explain both of these phases. And so We already knew that the infected cell death was what we needed to target because that's what drove the end of the infection. And so we just went in with a small modification to the equations and made that clearance of infected cells now dependent on the density. And so this gives you this really nonlinear clearance rate, where when you have very few infected cells, you get very fast clearance. And when you have a lot of infected cells, it really slows down. And that's what creates these two phases here. And this just shows a single parameter sensitivity, where that particular clearance rate really then still drives the back end of the infection and the full clearance of the infection. So we know that these things are really interacting with the host response and really driven by the host response. So early on in the infection, you have innate responses like macrophages and neutrophils. And then later on, you're really reliant on adaptive responses like the CD8 T cells. And then after that come the antibody. And of course, these two are really important for the next time that you get an infection. And so for today, we're really just going to focus on the CD8 T cells and looking at this really rapid decline that happens at the end of the infection. And so we developed another model that built upon that original smaller one, but included now CD8 T cells. So on the left-hand side, you can still see our target cells are becoming infected, undergoing that eclipse phase, producing more virus. But then the CD8 T cells are coming in, they're expanding, and then they're going to kill off those infected cells. And then a proportion of them are going to go on to generate memory and long-lived memory, so that when you see the infection again, you can take care of it. So some of the things that we found from this model is that the model fit our data very well. We have now two phases from these two equations here. One is just constant clearance, which gives you this really flat middle part, and then of course the clearance by the effector CD8 T cells that really drive that down. So with the effector response, we actually have a little bit of a blip early on, which we call infiltration, and then the huge expansion that we're really expecting to have the biggest effect on bringing these viral loads down. It occurs right at the time, or peaks right at the time when virus is fully cleared, right at 8 days. And then you can see how much higher they are after the infection is over than they were at the beginning, and those are really your memory cells. So we did a little bit of an experiment in the meantime to make sure that we were interpreting our model correctly and actually prove that it's right. So in this particular experiment, we were looking for which of these cells was actually in the lung tissue versus coming into the tissue and in the vasculature that's in the lung. So we used a technique where we injected an antibody retroorbitally right before harvesting the lungs. And then we used a larger flow cytometry panel to get out different phenotypes of these CD8 T cells. And what you can see here on the right is what we fit our model to was these data in the black squares. And what our model had predicted for the effector response is in the gray. And so that's not a fit, that's just an overlay of the model with the data. And so we can tell that what we had predicted to happen is actually what is happening. And we took it a little bit further and looked at both select phenotypes of effector and memory. Here we refit our model. The data look really similar to these, so the model fit perfectly. But so we can make some other conclusions about what certain phenotypes are doing and not doing. And I'm not going to talk too much about that today. It gets into some pretty nitty-gritty immunology. But at the end of it, we feel very confident that our model is working and that it's working well. So we did some ex silico experiments looking at just what is the critical number of CD8 T cells that are needed for resolution. So we know from doing these sensitivity analyses that if we change even just a single parameter to where we either get a whole lot more or a whole lot less CD8 T cells, we can change the clearance of the infection. So you can see how far that goes way up here in the red and the orange, but you don't actually get a huge benefit in clearance. And so that could actually lead to some really detrimental effects. Whereas if they're too low and they're sort of this critical level, then you get a really extended infection. And you can see that in these top ones a little bit more, where we plotted the maximum number of the CD8 T cells over the infection duration. And the duration doesn't change very much until you hit this critical level. And then all of a sudden you go from a 10 or 12-day infection out to a 30, 40, 50-day infection. And some of those longer infections are really seen in children, in some elderly cases, or in immunocompromised individuals. So the next experiment we did was really to test this and ask whether or not our model was predicting this correctly. So it's really hard to go in experimentally and change rates, because you can't really isolate a rate of a certain cell type, but you can change its population. And so that's what we sought out to do. We went in and depleted CD8 T cells. So we depleted them before we ever gave the infection. So by day 0, you're already a couple logs lower with your CD8 T cells. And then we depleted a couple more times at right at the time of infection, at 3 days after, and at 7 days after. And then looked at the response that we got. And there was a couple things that happened, and some of these are more interesting experimentally, in that we need to understand them so we can interpret our data correctly. And then some of them, of course, have more meaning clinically. So one of the things that we noticed is that the whole infection started from a different place. So like from the first get-go, we kill off all the CD8 T cells, but that creates cell death, which means other cells are going to respond to that. And ultimately, what our model suggested is that you actually have a decrease in the number of target cells. So you already have a little bit of protection. You end up with fewer cells being infected, which gives you, at this second day of infection, lower viral loads. So that was a little bit completely unexpected, but we needed that knowledge to really understand and interpret our model appropriately. And so you can see how these stay low throughout the infection. And then, of course, later on, when you expect the CD8 T cells to be there, they're not in sufficient numbers at day 8. But the last time we depleted was at day 7. So by day 10, there's enough cells there to clear out the infected cells, and the virus clears still by 10 days. And so we had a lot of interesting things that came out of this. We already knew that this particular depletion antibody can change both the phenotype and the behavior of the CD8 T cells, and that's actually exactly what our model predicted. So we had this decrease in the number of target cells, and then the only two parameters that it affected were the CD8 T cell influx was decreased, and that what leads to, in these first couple days, to the lower load or the lower levels of CD8s. And then we actually had an increase in the expansion, which is really trying to compensate for the reduced CD8 T cells. But nevertheless, our model was able to fit the data and say something meaningful biologically that both our data suggested as well as other people's data have suggested. Okay, so we're still feeling good about our model. That CD8 depletion data also suggested that the clearance rate is really dependent on the density. If we look at this on a surface plot here, now we have the infected cells against the CD8 T cells. As you can imagine, with a lot of infected cells but no CD8s, you're not going to clear very fast. And as the CD8s increase and start clearing, you sort of zip up to the top of this surface, and it gets very, very rapid. And so we put little markers on these figures just for illustrative purposes. So you can see how fast the infected cells are coming down, but your CD8 T cells are actually not changing that much. So you just need a few more to get that robust clearance. And it's a really dynamic system, where you have the infected cell numbers changing and the CD8 T cell numbers changing, and those two things are happening simultaneously. And there is some biological evidence of this density dependence. This is also a really hard thing to show experimentally, but between what we had found and some other biological evidence where there's spatial limitations of CD8 T cells moving around, they do take time to actually kill an infected cell, and they use a cooperative effect where it takes multiple interactions to actually kill an infected cell. So we don't think we're too far off with assuming that infected cells are clearing differently depending on how many of them are there versus how many CD8 T cells are there. So we did set out to look at this a little bit further too, and actually look at what the infection looks like in the lung and how many infected cells there actually are and what that actually means. And so we used an experimental technique called quantitative histomorphometry. It essentially takes a histologic image of the lung and stains for a protein of influenza called the nucleoprotein, and it'll stain all the cells that are expressing this protein, which means it's staining all the cells that are infected. So to be able to quantify this, we had a board-certified pathologist analyze these images, draw red lines and areas around the infected parts, which are these darker areas here, which are the ones stained with the antibody, and then this green outline of the whole lung to get a comparison of the infected area to the total area gives you a measurement of infected cells versus healthy tissue. And for the purposes, visual purposes, we color these in so they're a little bit easier to see. So we did this across the entire course of an influenza infection. Now, of course, these are different mice, so try not to let your eye follow the spatial aspects, but you can get an idea for where the infection is located, how big the lesions are, and how much of the total lung is actually becoming infected by the virus. Now, keep in mind, we use a sublethal dose, so no animals in our study would have died from the infection. They will all clear it perfectly in that 8 days. But what we can see here is that day 1, there are infected cells. There's just not very many of them. But by day 2, you're starting to see these lesions pop up. Now, remember with the viral loads peak at day 2. So you have very, little hot spots of the virus starting to pop up, but they are pumping out virus very rapidly, and you're already getting to a peak level. But what you notice between days 3, 4, and 5 is that area continues to grow. And this was at a time when the viral loads were actually not increasing. So your disease is getting worse, but it almost doesn't look like that if you're only focused on the viral loads. And then by day 6 is the first time we started to see some clearance. And so you get mixed areas of active infection happening along with clearance. And by day 8, that's when all the animals cleared the viral loads. You see no or very few infected cells. And then you can still see where the infection had been because there's so much destruction. And all the way out to at least day 10. Obviously, this could have went longer, but we stocked our data at day 10. And if you notice, this is that CT image I showed you earlier, where you have infection on both sides of the lung, you have large amounts of the infection, and that looks a lot like these CT images from infected patients. And quantifying that looks, pretty much like these images do, where the red is the active infection, where it starts to increase. It increases most rapidly between 2 and 3 days, kind of slows down a little bit, all the way up till 6, and then drops rapidly from 7 to 8. And that's when the inactive lesions in these green, where the infection is clearing and those come up. This blue line is just the addition of those two and the total lesion. So we went into our model and just asked if we can estimate this, like does our model do anything or say anything about these data? And what we found is if we take the cumulative area under the curve of the infected cells, then we can essentially add up all of these areas. And so this figure here shows that that's exactly it. On the left-hand axis is the percent active lesion, and on the right-hand side is the cumulative area under the curve. And so these are just overlaid. It's not a fit at all. It's just an overlay of the data. And our model suggests that we can capture these dynamics pretty perfectly, which was a real highlight for us. And we can do the same thing for the inactive lesion. So we're pretty sure that CD8 T cells were responsible. And what we found is that the inactive lesion can actually be predicted not by the CD8s themselves, but by the relative CD8s. And that's a little bit of a fine point, but it is important that it's not, you can just look at the CD8s that are there. There still is this calculation you need to do to look at the percent that is there. And it overestimated a little bit in the beginning. I think all that means is that the cells that are responding early on are not really involved too much in infected cell clearance. But they might be, and our histomorphometry just might not be picking up small changes. So if we add these two things together, we can get to an estimate of this total lesion. We thought it was nice that we can get the heterogeneity and kind of actually overestimate a little bit, which I think is better than underestimating. But our model does a pretty good job now of capturing the virus dynamics, the CD8 T cell dynamics, the infected lesion, the inactive, previously infected lesion, and then of course adding those two together to get the total. So one of the other things that we noticed was that these total lesion dynamics looked really similar to what our weight loss looked like. And we were mostly, focused on that you get a little bit of a change all the way up to 5 days, and then this spike at 6 that comes back down. So it doesn't take, too long to look that these might actually be related. So if you plot them against each other, where you have the weight loss on the X and the total lesion on the Y, you have this beautiful nonlinear increase that maybe gets a little less supportive once you get down to some higher weight loss. But this increase up, I thought was really neat. And so all we did was fit a Hill function, which captures this nonlinearity and gives us that relation from the total lesion to estimate weight loss. So what we do know is that inflammation also plays a huge role. And inflammation is a little bit distinct from the lesions that I just showed you. So of course, everything is related in some way because it's all happening during the infection. But inflammation is scored a little bit different. So our pathologists also scored inflammation, both alveolar inflammation, which is what's happening sort of in the alveoli of the lung, and then the interstitial inflammation, which is more of what's happening in the tissue. And those are in the white triangles and the yellow triangles. So you can see they're pretty close to each other, but there are some minor differences here and there. If you just plot these two things together, they look like they're probably not related, right? Inflammation comes up much faster. So by 3 days, you're already at a really high level of inflammation. It kind of flattens out, but by the end of this 10 days, you're not actually getting resolution of the inflammation, whereas you are getting resolution of the infected lesions. And so if we just plot these two things together, the total lesion against the inflammation, you can see that they are maybe somewhat correlated, but it does seem to be a little bit nonlinear. So there is a relation there, but it seems to be independent from what's happening on more of the tissue injury from the virus infection itself. But if we look again at whether or not these things can produce weight loss, so plotting them against each other maybe is a little bit misleading, because when you plot them with the weight loss on the X again and the inflammation on the Y, you see this beautiful and more perfect, especially towards the higher ends of the weight loss, correlation between inflammation and weight loss, and for both the interstitial inflammation as well as the alveolar inflammation. And so I put back on here the correlation with the lesion. And one of the things to notice here are these Hill parameters. So this N, which is the exponent here, tells you how nonlinear it is, how fast it's going up. And for the lesion, it's at 5, whereas for the inflammation, it's at 3. And so that tells us something. And then this K tells us at what point is that happening. So for the lesion, it's at about 3, 2.5 to, starting to get up towards 3% weight loss when you go up. But for the inflammation, it happens just a little bit earlier, right before you hit about 2% of your weight loss. You go sharply up in the inflammation. And so we thought that we could probably also model inflammation. So we know that inflammation is caused by several different things, most predominantly macrophages and neutrophils, but we didn't want to model those cells or other things that cause inflammation because while we could do it and we probably will do it at some point, it gets awfully complex to say what is actually influencing inflammation. So we wanted to kind of pull back and absolve ourselves from having to make those kinds of claims and just ask, can the infected cells reproduce this inflammation? In the sense that infected cells produce cytokines that bring in these cells like macrophages and neutrophils. And so we just built this really small, straightforward equation, having contributions from both infected cell classes, and we get a pretty nice fit to these data. And what this suggested actually was that first class of infected cells before they're producing virus actually have a stronger effect. And so that's distinct from the histomorphometry I just showed you in those lesions, which are really driven by the post-eclipse phase cells that are actually actively producing virus. All right, so hopefully I have taken you at least a little bit through this and shown us how we can get from viral loads all the way down to disease severity, but that it's not a straightforward path. And that I think is really important, but also gives us a lot of insight in how we might be able to use some of these data to say more about the infection, particularly if we don't have everything in the middle. But I'll just walk you through it quickly. Just as a summary, we took, perhaps starting with viral loads, we used our kinetic model to predict CD8 T cell dynamics as well as infected cell dynamics. Those things can get us some estimates of the lung injury as well as inflammation. And then once we have the total or the lung inflammation, we can estimate disease severity from there with this nonlinear Hill function. Okay, so I'll end just a couple notes about antiviral therapy, which I told you earlier, is really only effective early on in the infection. You can see here back at this first influenza study that I showed you, they had some data where the individuals were treated with oseltamivir, which is Tamiflu, at different times. That's all these markers that are here in the middle. There's different efficacies. Their model did an okay job of capturing it, but obviously not great. But there also wasn't a lot of data to say much about. We did it in mice and using a collaborator's data, where we tried to simultaneously capture whether treatment was initiated early at day 0, so more prophylactically, versus really late in the infection at 5 days. And there's only a small difference between this blue and the black line here, and that's because at late in the infection, right, where you're past all of that virus life cycle phase, you don't get a lot of efficacy of the antiviral. Whereas early on, you can really pull down those viral loads. And so what our model suggested to be able to capture these two things simultaneously is that we have an effect on production because the oseltamivir is inhibiting neuraminidase, which is really important for virus exit out of a cell. But where the stronger effect was what we call the protection of the target cells. And what that translates to is a little bit unclear, but more of just like removing the pool of target cells from being able to become infected in the first place. And that was really the only way that we could capture both of these data sets simultaneously. And now that we have this histomorphometry, it makes complete sense. So here are a couple of images. These are not our data. These are from a separate study that looked at oseltamivir, and they've done some other nice antivirals and combinations of antivirals. But where you get the huge benefit of these treatments is actually in reducing the lung pathology. So you can really see the reduction in the area of the lung that becomes infected. And that's going to decrease your symptoms. It's going to decrease your inflammation, right? And so if we think about that disease severity, of course, it's going to decrease your disease severity when you go from, say, in this one, they had almost 98% of the lung infected down to maybe, 72 or something lower. And of course, if you treat at different times, you can get larger efficacy from these antivirals. But this really gave us a lot of insight by going through this and really trying to connect what's happening with the viral loads and the lung pathology to what it also means with disease severity, so we can better interpret antiviral efficacy for one, changes in immune response, or other modulations that we might see just from natural heterogeneity, either from the pathogen or from different humans and how they respond to the viruses. So I will end there and take any questions that you might have.
Thank you, Amber, for a very interesting webinar. We do have some questions. The first has to do with the viral load. And it says, much of the viral dynamics profiles are plotted with viral titers. How is the viral titer measured? Does it measure the viral infectivity using a plaque assay or based on a quantitative PCR viral load?
So we use either TCID50, which is a tissue culture infectious dose, or on occasion we'll use a plaque forming unit. So those get you the infectious titer, whereas RNA will get you potentially non-infectious particles or just our RNA that happens to be laying around. We find that a little bit less accurate just because you can still have a lot of RNA there, but no actual infectious particles. And so most of, on both of these graphs that you can see here and in everything that I showed, it's all TCID50.
Given the difference in similarities between influenza and SARS-CoV-2, how much of this model and mechanisms could be translated to COVID?
Yeah, that's a great question. So we're working on it. I think in general it probably will be able to. The big differences is that COVID acts on a different time scale, so it has a little bit longer of an incubation. And it has these really interesting tricks up its sleeve with different host responses. And so it alters them in a way that, let's say, to focus on the CD8 T cells here, a lot of people, as well as we see this in animal models, we end up with the CD8 T cells being depleted. And so, of course, that's going to lead to potentially longer infections and more pathology. So, yeah, there are just some different similarities, and I'm excited to get our data and actually analyze it and try to understand it. The mouse models are not as developed and easy to use, so we have a little bit of a complication there. But a lot of the disease, I think we're going to find a lot of similarities on that front, with adding in these tricks that COVID does to really change the innate responses that then, of course, lead to the changes in the adaptive responses. So fingers crossed that it will, but honestly, it's really interesting to find the places where your model breaks down So there's a small part of me that actually wants that to happen so that we can learn even more and try to find the similarities between.
Okay. And have you considered the use of perhaps a mixed effects modeling approach to estimate inter-individual variability on some of the parameters or processes in order to distinguish between subpopulations or establish covariates explaining the variability?
The short answer is yes. So with our influenza data, we have the unfortunate ability that we can't track individual animals. And so on each one of the time points that we have data, it's a destructive sample, which means that it's really hard to connect them and say something about the heterogeneity. But where we have done it is in a sort of a similar infection with para-influenza virus. And so in an experimental system within para-influenza, there's a nice bioluminescent virus that you can use imaging to track the infection and then correlate that to viral loads. And that has given us a little bit of an ability to do that, at least in an experimental system, and start to understand where some additional heterogeneity might be. What we found from that with, it's a little bit tricky with animals because they're supposed to be the same. And you're supposed to have minimal heterogeneity to have, reproducibility. But what that study showed is the sensitive processes that we have found in these other models, just from doing those one parameter sensitivity analyses, are the ones that show up as being sensitive and having the heterogeneity when you do have individual animals.
You mentioned your measure of inflammation in the model is perhaps the chemokines or the attractants for the immune cells, and yet cytokines are known to cause weight loss. Have you looked in your mice to see if you've modeled one or the other or both of those accurately?
We have looked a little bit, so we actually have all the data for the cytokines. At a first glance, we haven't noticed anything that perfectly correlates necessarily, but the inflammation dynamics, which look a lot like the macrophages, they look a lot like the neutrophils. They also look a lot like IL-1, IL-6. Those are probably the two that they're most related to, which also makes sense. Those are two highly inflammatory cytokines that also bring in, things like macrophages. So I don't know. I guess the short answer is sort of yes, that we have, but we kind of take a step back and try not to insert too much of what we think happens when we can't say a whole lot dynamically with our model.
Can you comment on what your model would suggest as an explanation for chronic infection?
You mean like chronic with, say, HIV or like a longer, say, influenza infection?
I think a longer influenza.
Yeah, so I don't think we would ever call it chronic, but there are extended influenza infections. So I think a couple things are potentially happening. One is the one that I mentioned earlier, where you have either immunocompromised patients or children that don't have robust immune responses. And they, depending on the age of the child, they might not even have a very well-developed CD8 T cell response. And so if you don't have those things controlling it, you're going to end up having a longer infection. Where we do see longer, maybe disease courses, where you might actually clear the infection, I think is more from this lung pathology and from secondary infections. So a lot of people come into the hospital after they have even maybe started feeling better from the flu infection, but get a secondary bacterial infection after that. And that is what it will cause them to be hospitalized, and it really extend out what would be an influenza-associated disease.
Have you modeled a dual infection?
I sure have, yeah. So I did a lot of this in my graduate work, and we're still doing a lot of it now. But yeah, we developed a model to try to understand pneumococcal infections after influenza, and we found some really interesting dynamics. So influenza, at least in our system, depletes the alveolar macrophages. So your macrophages that are resident in your lung, they're really supposed to be there to take care of things that come in. And throughout the course of the influenza infection, they become depleted, which allows bacteria to come in, very little bacteria to come in and establish. And then the immune response just goes haywire. And you get this huge neutrophilic influx. And at least in our animal models, it's usually 100% lethal within a couple days.
So somebody asks about the particular images you're showing. It says, to my eye, the left lung had more lesion or infection, especially in the inactive phase of your histopathology data. Do you think surface area versus volume of the different lobes may play a role in the infection response kinetics? And could your models be adapted to consider spatial heterogeneity of infection and lesion formation?
Yeah, that's a great question. So some of it boils down to the infection itself. We can experimentally control it a little bit how much gets onto one side or the excuse me, one side or to the other, just by kind of tilting the mouse to one side. We really actually tried to do that specifically for this because we wanted that even distribution. And of course, that's not always going to turn out to be even, but that's why we see it on one versus the other. Why some mice get it into the lower, some of the lower areas or more on one side or the other, I would say we don't know. But it could, but the different sizes of the lobes, the different, architecture and structure, and maybe, how well it actually got in there and where it initiates from, I think all play a huge role. We are now trying to extend this into a spatial context and working on first adapting ODEs into spatial modeling, and then trying to sort of grow up from that. And so hopefully that'll give us a little bit more insight into it as well.
That leads to the next question, which is regarding the differential equations for your mechanistic models. Is that published?
It's on bioRxiv right now. We've been under an extremely long review process where we had to do all those extra experiments, but hopefully, and in the next couple of months, it'll be published. But it is, most of it is we've put on bioRxiv.
Great. I think we've gotten all the questions, and so I'd like to thank our speaker today, Amber. It's been a very interesting and I think very informative webinar for our audience and myself. And I'd like to thank you very much for giving it. And I hope that you and everyone else will come back for a future webinar.
Yes, thank you very much.
Thank you. Have a great day, everyone.